Hi, I'm Lisa Metzger from the Psychs Lab, and today I'm going to be talking about the potential role of the transcription factor, Upstream Stimulatory Factor, or USF, in the DNA damage response. A staggering 1 in 50 people will develop a non-Hodgkin lymphoma at some point in their lives. At least 60% of these cases are considered to be aggressive, causing this to be the sixth most fatal cancer type in the United States. But despite its prevalence and severity, it remains difficult to detect early and with few definitive signs. Our research focuses on a potential biomarker of USF to help diagnose or recognize risks of developing non-Hodgkin lymphomas. This cancer can form at almost any stage in the B-cell life cycle, from the accumulation of mutations caused by a number of factors causing DNA damage, ranging from chemical carcinogens to radiation to infections from pathogens like Helicobacter pylori or the Epstein-Barr virus. Cells have extensive and tightly regulated DNA damage repair pathways like the p53 pathway, but they can be error-prone. These errors, especially when cells undergo frequent or extensive damage to their DNA, accumulate as mutations and lead to oncogenesis. These cells in particular inherently are exposed to DNA damage as double-strand breaks are formed during their affinity maturation process, creating highly specific antibodies, so they require a strong DNA damage response. It is known that the p53 damage response pathway has high levels of protein regulation, but it's still unclear whether transcriptional regulation also has a heavy role. Our preliminary data has shown that USF has bound to the p53 promoter, and based on ENCODE data, there are putative binding sites for USF at the p53 promoter. Previous studies have shown interactions between USF and p53 at the protein level as well. We created USF knockdown cell lines using RNA interference and studied the long-term, 7-day effects of DNA damage, and found that there was a high level of gene dysregulation unique to our USF knockdown cells, many of which these genes were involved in DNA damage repair pathways. Literature also has shown that a lack of USF correlates with higher risk of cancer. We are working to determine the role of USF in regulating p53 and other aspects of the DNA damage response pathway to elucidate whether this transcription factor could be a potential intervention point in non-Hodgkin lymphomas, as a biomarker indicating early stage cancer, or even as a potential target of gene therapies.